Bismillahirrahmanirrahim, and welcome back to this next video. And in this video and the videos to come, I'll be focusing on the isotypes of the antibodies. If I summarize the whole story that we have discussed so far about the antibodies, is I've told you that antibodies, they're also known as the immunoglobulins. They are glycoproteins in nature. They are produced by the plasma B cells, and they are part of the adaptive immune response. The antibodies, they are responsible for recognizing and neutralizing the foreign antigens like the bacteria, the viruses, the fungi, and other harmful particles like the toxins produced by different pathogenic organisms. We discussed about the structure of the antibodies, and I've told you that the structure of an antibody consists of two heavy chains and two light chains. And in the structure of the antibody, you are going to see the variable regions which are responsible for binding to the antigens, and you are going to see the constant region that interact with other components of the immune system like the complement system. Now, we usually classify the antibodies into five major isotypes. They are also known as the five major classes of the antibodies. So, the isotypes of the antibodies and the classes of the antibodies, they are one in the same thing. And these five major classes of the antibodies, they have distinct structural and functional properties. Now, what is the basis of classification of antibodies into these five classes? So, the basis is the differences in the constant region of the heavy chains. So, these isotypes, they are actually classified based on the differences in the constant region of the heavy chains. We are going to start our discussion with the first class of the immunoglobulins, and that is known as the immunoglobulin G. Now, this immunoglobulin G, this is a monomeric antibody. It consists of two heavy chains and it consists of two light chains. And the immunoglobulin G have a classic Y-shaped structure, and this is the classic Y-shaped structure. And if you look at this, you are going to see two light chains. This is one light chain. This is another light chain. So there are two light chains in the immunoglobulin G. Similarly, there are two heavy chains in the immunoglobulin G. So this is one heavy chain and this is another heavy chain. Now, the structure of the immunoglobulin G, it is actually a tetrameric quaternary structure, and it is approximately 150 kilodalton in molecular weight. If you look at over here, this particular region of the antibody, that is known as the hinge region, and this hinge region is actually responsible for making the disulfide bonds, and this disulfide bond that you can see over here, that is actually connecting the two heavy chains of the antibody to each other. Now, both of the forks of the Y-shaped antibody have an identical antigen binding site. If you look at this particular region over here, this is actually the antigen binding site. And if you talk about the immunoglobulin G, so both of these antigen binding site, they are identical to each other. That means they are going to bind to the same antigen. Now, why we call this as the immunoglobulin G? Because if you look at the heavy chains of the immunoglobulin G, they are known as the gamma heavy chains, and this G actually refers to the gamma, and this gamma is actually representing the particular heavy chains that are present in the immunoglobulin G. If you look at the molecular weight of the gamma heavy chain, that is approximately 50 kilodalton. So now you can calculate the molecular weight of the immunoglobulin G over here. This is 50 kilodalton. Another heavy chain is 50 kilodalton, and these light chains, they are actually 25 kilodalton. So 50 plus 50 is 100, 25 plus 25 is 50. So you are actually going to get a molecular weight of 150 kilodalton for the immunoglobulin G. Now, there are two more terms that you usually see when you talk about the structure of the antibody. One that is known as the FAB. We have already discussed that in detail, but there is another term that you usually see when you study about the structure of the antibody, and that is known as the FV. So this FV, which is known as the fragment variable, This is actually the smallest antigen binding unit of the antibody, consisting only of the variable regions over here. So if you talk about this variable region over here of the light chain, this variable region of the heavy chain, similarly this variable region of the heavy chain, and this variable region of the light chain. So these particular structures, they are known as the FV. When you talk about the FAB, which is the fragment antigen binding, so this is actually the FV plus part of the constant region of the light and the heavy chains. So that means that the FV is a smaller structure, and the FAB is actually consisting the FV plus some part of the constant region of the light and the heavy chain, and these are actually the constant region of the light chain, and this is the constant region of the heavy chain. So this is one of the differences that should be clear in your mind. If you talk about the immunoglobulin G, these immunoglobulin G, they are actually the most abundant antibody in the bloodstream, and they actually account for about 75 to 80% of all circulating immunoglobulins in your serum. So the IgG, the immunoglobulin G, they are the most abundant antibody. Now, these immunoglobulin G, they are further divided into four distinct subclasses of the immunoglobulin G. One is known as the immunoglobulin G1. The other one is known as the immunoglobulin G2. The third one is known as the immunoglobulin G3. And the fourth class of the immunoglobulin G is known as the immunoglobulin G4. Now, when you talk about these subclasses of the immunoglobulin G, so they are primarily based on differences in the constant region of the heavy chain. So, as you are talking about the subclasses of the immunoglobulin G, so you are going to focus on the gamma chains that is present in the immunoglobulin G. So, this should be clear in your mind that we are making the classes of the antibody based on differences in the heavy chains. We are making the subclasses of the immunoglobulin G based on the differences in the specific heavy chains, which are known as the gamma heavy chains. So we are not talking generally about the heavy chain. We are talking about a specific type of the heavy chain, which is the gamma heavy chain. So the subclasses of the immunoglobulin G, they are based on differences in the constant region of the gamma heavy chains. And these differences, you can see a variety of them. You can see various differences between the subclasses of the immunoglobulin G. So one of the differences would be the differences in the amino acid sequence in the constant region of the heavy chain of each subclass. What that would mean is that there would be amino acid sequences differences in this class, in this class, in this class, and in this particular class. So this is one way you can see at the differences in the constant region in the of the heavy chains. Secondly, there are differences in the length of the hinge region, and the hinge region is actually the flexible region of the antibody that connects the FAB and the FAC region among the different subclasses. Now, another important difference between the subclasses of the immunoglobulin G, that would be the amount and the type of the glycosylation. You are going to see that variable between the different subclasses of the immunoglobulin G, and this amount and the type of glycosylation that influence the antibody interaction with the immune cells and the complement proteins. And another important difference between the subclasses of the immunoglobulin G, that would be differences in their biological properties, such as their effector function, like the complement activation, the antibody-dependent cellular cytotoxicity, and half-life in the circulation. So in the next video, we are going to see about the first subclass of the immunoglobulin G, which is known as the immunoglobulin G1.